Hello students, this is Mrs. Sonal Patil. In a previous class we have studied about Lacoperon concept. Today we will study about the Human Genome Project. The term genome was coined by H. Winkler to the complete set of chromosomal and extrachromosomal genes of an organism or a virus. So genome is nothing but the total number of genes located on a haploid set of chromosomes. So genome refers to the one complete set of genes for the species. We are aware that each human individual has a separate identity which differentiates him from other, and this identity is due to the presence of specific genetic makeup. The genetic makeup of an individual lies in the DNA sequence. These assumptions led to the discovery of complete DNA sequence of human gene. The genetic engineering techniques help to isolate and clone any piece of DNA, and availability of simple and fast techniques are used for the determination of DNA sequences. It led to launching of an ambitious project called as Human Genome Project. So Human Genome Project is a scientific project concerned with the study of base sequences of DNA molecules of complete set of chromosome. It was started in 1990 and completed in 2003. It is an ambitious international project which was started, I told you it was started in 1990 by molecular biologists to understand the genomes of humans and other organisms. It is designed to identify and locate 35,000 to 40,000 genes present on 23 pairs of chromosomes contained in the nucleus of the cell of humans. Initially, United States of America took the lead in Human Genome Project, and later 14 other countries set up their own Human Genome Project. These countries were Australia, Canada, Brazil, Denmark, France, Germany, Israel, Italy, Japan, Netherlands, Russia, Sweden, and United Kingdom. Recently, more than 1,000 scientists from 50 countries, including India, coordinate and monitor worldwide research under Human Genome Project. It is also believed that the Human Genome Project will give us insights into the fundamental mechanisms of life and led to an era of molecular medicine with precise new ways to prevent, diagnose, and treat diseases. Goals of HGP are identify all the approximately 20,000 to 25,000 genes in human DNA. Secondly, determine the sequence of 3 billion chemical bases. Store this information in databases. Improve tools for data analysis. Transfer related technologies to other sectors such as industries. Address the ethical, legal, and social issues that may arise from the project. So these were the goals of the Human Genome Project. Coming to the methodologies, there are two major methodologies used to study the human genome. One approach is identification of all the genes in the cell that are expressed as RNA molecules. This technique is referred to as expressed sequence tags. Another approach is sequencing the whole set of genome. Both coding and non-coding regions are considered, and later assigning different regions in the sequence with functions. This technique is called as sequence annotation. The commonly used host for sequencing were bacteria and yeast as vectors were called as bacterial artificial chromosome and yeast artificial chromosome. So here what was done, Human Genome Project followed the technique of sequencing the whole set of genome. For sequencing, the total DNA from a cell is isolated and broken into random fragments of relatively smaller size. These fragments were cloned in suitable host using specialized vectors. The common host, as I told, were bacteria and yeast, okay, which were called as bacterial artificial chromosome and yeast artificial chromosome. Recently, PCR, polymerase chain reaction technique, is applied for amplification of each DNA. So these were the two methodologies which were applied. Coming to the DNA sequencing procedure in HGP, the fragments were sequenced using automated DNA sequences that were worked on the principle of method developed by Frederick Sanger. These sequences were arranged based on some overlapping fragments for sequencing. Alignment of these sequences was humanly possible. Therefore, specialized computer based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and repetitive DNA sequences known as microsatellites. Silent features of Human Genome Project include important, will be asked in the exam. So this question on Human Genome Project goals may be asked or silent features may be asked, so important from exam point of view. The human genome contains 3164.7 million nucleotide bases. The average gene consists of 3000 bases. The largest known human gene being dystrophin at 2.4 million bases. The total number of genes is estimated as 30. 99.9% nucleotide base sequences are same in all peoples. The function of 50% genes discovered is unknown. Less than 2% of genome codes for proteins. Repeated sequences make up very large portion of human genome. Chromosome 1 has most genes and Y has the fewest. It is identified about 1.4 million locations where single base DNA differences or single nucleotide polymorphism occurs in humans. Application of HGP include it helps in diagnosing and treatment of genetic diseases. It helps in understanding functioning of genes and their regulation. So it is helping. So HGP helps in identifying the genes associated with various diseases. Example, cardiovascular disease, diabetes, arthritis, and to develop new effective gene therapy. Over 30 genes which are associated with the breast cancer, muscle disease, deafness, and blindness have already identified. Recent advances in genetics coupled with the data on Human Genome Project will help in modern medicine practice. The knowledge can be used to study mechanisms of drug action which will lead to a faster clinical trials. Human Genome Project resulted in the establishment of pharmacogenomics which evolved from pharmacogenetics. So these were the applications of Human Genome Project. Come to the next part, that is DNA fingerprinting. It is accepted the fact that except identical twins in humans, every living organism differs only due to nucleotide sequences of chromosomes. Since the nucleotides form codes, this coded informations can be profiled to produce most authentic identity card of any organism. The complicated technology that facilitates the identification of individuals as at genetic level is known as DNA fingerprinting. It is also called DNA profiling. The technique has been used solving disputed problems of parentage, identity of criminals, rapists, reuniting the lost children, identity of burnt or identified dead body, etc. It involves matching of a short piece of DNA of victim with the DNA of suspect. So if one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be daunting and expensive task. Imagine trying to compare two sets of 3 into 106 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in the some specific regions in DNA sequence called as repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk of DNA forms a major peak and the other small peaks are referred to as satellite DNA. Depending on base composition, length of the segment and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism arises due to mutation. New mutations may arise in an individual either in somatic cells or in germ cells. If a germ cell mutation does not seriously impair individuals' ability to have offsprings who can transmit the mutation, it can spread to other members of population. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant at the locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher as mutations in these sequences may not have any immediate effect or impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the basis of variability or polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large scale changes. The technique of DNA fingerprinting was initially developed by Alec Jaffrey. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats VNTR. The technique as used earlier involved southern blot hybridization using radio labeled VNTR as a probe. It included firstly isolation of DNA, second step digestion of DNA by restriction endonucleases, thirdly separation of DNA fragments by electrophoresis, and then transferring of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. Fifth, hybridization using labeled VNTR probe, and lastly detection of hybridized DNA fragments by autoradiography. So these were the steps which are included in DNA fingerprinting. The VNTR belongs to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The number of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of different sizes. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in population except in the case of monozygotic or the identical twins. The sensitivity of the technique has been increased by the use of polymerase chain reaction, that is PCR. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider application such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. So these are the steps which are being shown in the DNA fingerprinting. So it involves firstly the collection of the samples, then second step extraction of the DNA, thirdly separation of the DNA fragments, then extraction of the DNA fragments, then tagging of radioactive DNA probes, and last is autoradiography. graphy. So this diagram shows the steps which are involved in the DNA fingerprinting. Lastly, the applications of DNA fingerprinting. DNA fingerprinting technique is used to identification of criminals like murderer, robber, rapist, etc. It is employed in solving parental disputes. It is useful in identifying an reunion of lost children with their parents during natural disaster like earthquake, flood, tsunami, etc. It is useful in identifying the victims of plane crash, building collapse, fire breakouts, etc. It is used in population diversity determination. It is useful in the determination of genetic diversity and evolutionary biology. So these were the applications of DNA fingerprinting. With this, we have completed the chapter molecular basis of inheritance. In the next class, we will begin with the next chapter. Thank you.